Yeah. Dear students, next question, 16th question. Kinetosome is found in option A, flagella, option B, cilia, option D, villi, option D, C, villi, option D, plicae circularis. Kinetosome is found in. So for this, option is A. Answer A. But if this is the type of question, then the success of getting marks in such type of question depends upon the knowing the meaning of the word. Here, you should know the meaning of kinetosome. Kinetosome meaning, if you don't know, then it will be difficult to write the answer. Kinetosome means it is also called basal body. Kinetosome means it is also called basal body. I think some of you are still confused with this term, basal body. You might have seen that the structure of flagella. Flagella stands on a particular cell base that is called, this is flagella, of course, and it stands on a structure that is called basal body. Basal body. Okay, now I think it's clear to you. So kinetosome is also called basal body, and every flagella possesses one kinetosome. It is also called basal granule. Basal body is also called basal granule, or it is also called blepharoplast. It is also called blepharoplast. Blepharoplast. Okay. And what is shaft? Shaft is hair-like structure, projected part of flagellum. Hair-like structure, projected part of flagellum. Okay, hair-like structure, projected part of flagellum is called shaft. Then this consists of protein called dynein. What is the protein found in the shaft? That is dynein. Okay. Question number 17. Now let us move to the next question, 17. Which of the following is responsible for aging? Everyone is very conscious about age. So which of the following? Option A, nexin. Option B, desmin. Option C, vitmentin. Option D, peroxide. Out of this, which one is responsible? Let us see. Option D, peroxide. If you have strong enough peroxide, you will become young and energetic. So option D. Because peroxisomes helps in removal of toxic substance. Removal of toxic substance. Removal of toxic substances. For example, while consuming food, if there is a small trace of phenol, if it enters to our food, then that phenol is poisonous. Formaldehyde, if you are working in lab while preserving the specimen, if formalin touches to your hand, then while consuming accidentally, if it goes to your digestive system, it is removed by peroxisomes. And it is also helps to remove phenol, formalin, and other content like methanol and ethanol, etc. Okay. Then See, it's very interesting fact I want to tell you. During metabolism, molecular oxygen used by various enzymes, during metabolism. What is metabolism? Some total activity that takes place inside the cell. It may be constructive, it may be destructive. For example for constructive metabolism is joining of amino acids, formation of a long chain of amino acid is a constructive process. Breaking, for example, when you consume chapati, it should be broken down into amino acid. That is destructive process. Okay, chapati is a plant protein. Our body stores it by converting it into animal protein. So that you should remember. So here, dear students, what they are telling, during metabolism, molecular oxygen is used by various oxidative enzymes. Various enzymes, they use the oxygen, and like urate oxidase and deamino acid oxidase. It produces hydrogen peroxide. See, I will write. During metabolism, molecular oxygen is used by oxidative enzymes. Enzymes like I will mention few. Urate oxidase, and then amino acid oxidase, and then and both of these, they produce hydrogen peroxide. And what does hydrogen peroxide causes? It causes mutation of DNA. See how interesting it is. And this mutation of DNA is responsible for aging. So our food, if you consume less food, you will survive for a long period. If you consume more food, the aging will be fast, according to this theory. So what they say, during metabolism when it occurs, whenever we consume the food, molecular oxygen used by the oxidative enzymes. Oxidative enzymes, when they work, when there is a food, and for the work they require energy, so they consume oxygen, and it produces urate oxidase and deamino acid. Some enzymes like urate oxidase and deamino acid oxidase. These enzymes, they are responsible to produce hydrogen peroxide, and that mutates the DNA, and this DNA mutation is responsible for aging. That you should remember. Then nexin is a protein found in shaft. Keratin, it is found in hair. Desmin is a protein found in muscle. Desmin is a protein found in muscle protein. Okay, desmin is a muscle protein. Vitamin is a positioning of nucleus. Vitamin filament. Positioning of nucleus in the cell. Okay, so this is with regard to question number 17. I'll move to the next question. Question number 18. What is the question? What they asked in question number 18? The question is, which of the following set represents pigments of mitochondria? Cardiolipin and anthocyanin, anthoxanthin and chloramphenicol. Option C, anthocyanin, anthoxanthin. Option D, cardiolipin and chloramphenicol. So the answer for this is C. 18th question, option C. Answer is C. The color of anthocyanin, depending upon its concentration, it will be red, purple, blue. Anthoxanthin, color is ivory to deep yellow. Ivory to deep yellow. Okay. Then cardiolipin. It is more in mitochondria. Cardiolipin is more in mitochondria. Then chloramphenicol, antibiotic, inhibit mitochondria protein synthesis. Chloramphenicol. It's an antibiotic. A person who takes antibiotic will be tad. That tadness is mainly because some antibiotics will prevent the mitochondria to produce the proteins, will disturb the normal work of the mitochondria. When mitochondria is a powerhouse, when it is not able to do the work, then the person will become weaker and weaker. So mitochondrial protein synthesis, mitochondrial antibiotic inhibits mitochondrial protein synthesis. So this is about question number 18. Now I will move to the next question, that is question number 19. What is true about fluid mosaic model? Option A, phospholipid layer is sandwiched between two protein layers. Phospholipid monolayer is present on the top of the protein layer. Phospholipid layer present on the top of the protein layer, and proteins are embedded at places in the phospholipid bilayer. So the option is D. I won't explain this because you know the structure. I'll just directly go to the next question. Which of the following is correct for prokaryotic ribosome? It is option A. Because you know that eukaryotic ribosome, 80S type. Eukaryotes, 80S ribosomes, that is larger subunit is 60S and smaller subunit is 40S. Then prokaryotic ribosome is prokaryotes. It is around 70S, where the larger subunit is 50S and smaller subunit is 30S. Okay? Smaller subunit is 30S. So you should remember this. Next, question number 21. Question number 21. In question number 21, There is a diagram. Different types of meiosis are given diagrammatically and labeled with alphabets. There are 4 options. Choose the correct option. So I will directly go to the answer here. Answer is C. So there are different types of meiosis. One is gametic meiosis. Gametic meiosis during gamete production. During gamete production. Gamete formation. Okay? It occurs during gamete formation. Second one is zygotic meiosis. Zygotic meiosis. Zygote formation. Zygote formation. And then sporic meiosis. It occurs during spore formation. It occurs during spore formation. And meiocytes are the sites where meiosis takes place. Sites where meiosis occurs. They are the sites where the meiosis occurs. Then I will move to the next question. Question number 23. 22. In which stage of the prophase first? In meiosis first, synthesis occurs. Leptotin, pakitin, diplotin, zygotin. So it is option D. Option D. Here I know synthesis, the word. Synthesis is it's not a complicated. It don't have any complicated meaning. Synthesis means synapses. Synapses, you know. This word is familiar to you. Synthesis is another word for synapses. That is pairing of homologous chromosome. Leptotin is called leptotin because leptotin means thin thread-like. Thin thread-like. Leptotin. Pakitin. It is a synapses formation. Synapses formation. And diplotin, pairing of homologous chromosomes, tetrad formation. Tetrad or bivalent formation. Bivalent formation. Then diakinesis. Diakinesis. In this terminalization of chiasma. Terminalization of chiasma. So these are all some of the important aspects that you should remember along with the answer. Now I'll move to 23rd question. 23rd. Question number 23. What they asked, in which stage of prophase first, okay, 23, which of the following is unique to mitosis and not a part of meiosis? Homologous chromosomes behave independently. Chromatids are separated during anaphase. Homologous chromosomes pair and form bivalent. Homologous chromosomes cross over. So, in this, you should find out following unique to mitosis. That means which occurs only in mitosis. It is obviously option A. Question number 24. 24, what they are asking? Choose the diagram which is correctly shows the anaphase first stage. What are the characters of anaphase first? Chromosomes. Meiosis first is reduction division. So chromosome should be there in the opposite pole. So it is option D. Option D. Question number 25. In the G2 phase of the cell cycle, the characterized by the presence of, it is option D. 25, option D. You know, G2 phase is in between G1 and S phase. S means synthetic phase. Now, question number 26. The major event that occurs during the anaphase of mitosis, which brings about equal distribution of chromosomes. Question is, the major event that occurs during anaphase of mitosis, which brings about equal distribution of chromosome is, you know, mitotic division is equational division. In that, option A, replication of genetic material. Option B, splitting of chromatids. Option C, splitting of centromeres. Option D, condensation of chromatin. So, by observing those, what event that helps to equational division? It is splitting of centromere. That means instead of chromosomes move to opposite pole, chromatids, daughter chromosomes will move to opposite pole. So it is option C. Option C. Then 27, question number 27. Here there is one structure has been given. So what the question? Which of the following structural formula represent maltose alpha 1,4 linkage? So for that question, option is answer B. Answer B. So A, which structure? It is sucrose alpha 1,2 linkage. A, sucrose alpha 1,2 linkage. 1,2 linkage. And B is the answer. It is maltose alpha 1,4 linkage. Now I will go for C. Lactose beta 1,4 linkage. Lactose beta 1,4 linkage. Then D, chitin. D, chitin. So the structure of A is sucrose alpha 1,2 linkage. B mentioned in the answer. C, lactose beta 1,4 linkage. D, chitin. Now 28, question number 28. It's match the following. So in that match the following, the option is A. 28th answer is A. Now I'll move to 29th question. 29th question. Question number 29. Question number 29. Which of the following vitamins are produced from intestine? It is option B. Option B. That is the vitamin was coined by, the name vitamin was coined by Funk. Coined the term vitamin. Vitamin. And dear students, vitamin D and A and K are produced in vitamin D, A and K are produced in liver. You should remember this, liver. And vitamin D is also produced in skin. And fat soluble vitamins are A, D, E, K. You can remember it as ADK. So A, D, E, K. Okay. So now I'll continue this in the next part, that is question number 30.